Hi, I'm Dr. Lawrence Swalikoff, naturopathic physician. Joining me today is Franco Cavallari, a biomedical researcher from Biologics Pharmamedical. Today we're going to discuss some new interesting technology and research called BioBDMC50 technology. How are you doing today, Franco?
Good, Lauren. Thanks for having me.
Great. Before we jump into this curcumin research and the new product CURCUMIN III that we have available, tell us a little bit about how you got into natural research, what Biologics is doing nowadays with their research. Fill us in a bit.
Well, Biologic Pharmamedical has been focused on studying natural compounds the same way the pharmaceutical industry studies drugs. And the key reason why we engaged and ran down this path is because we see a lot of very effective natural compounds, many of which are polypharmacological in the way they deliver their activity, meaning there's multiple constituents delivering the pharmacology, but also multiple points of drug activity in our cells with these botanicals. And so it's kind of the natural philosophy, the holistic philosophy is a little bit different than that of the allopathic medical practitioner, whereas the allopathic medicines are studied in the context of selectivity to be extremely particular and pointed in their activity, whereas we see with this holistic approach, the polypharmacology is said to have a facilitative result of the biological system as opposed to a targeted drug target that we see in allopathic medicine. And both philosophies have a place. What I decided to engage in is to actually study the holistic approach by isolating each of the components in these holistic medicines and then studying them individually so that we understand better how each of these constituents can contribute to the polypharmacology, and then be able to re-engineer them if needed to be more selective for an indication. And we've been very successful with many drugs, including our natural drug from the curcumin extraction. And curcumin, has become a focal study for us because we know that inherently there's a potent anti-inflammatory activity for curcumin, but I began to study it a little bit differently than how it was typically studied, because I myself also am one who suffers from an autoimmune condition that I found was treated very effectively when I used engineered versions of the curcumin. And I found that early on, upon using curcumin to help treat the symptoms of my condition, I found that one curcuminoid in particular, when used properly in conjunction with the other curcuminoids, had an extremely potent effect on the resolution of the symptoms that I was experiencing with ulcerative colitis. And that then led to the investigation that has taken me down about an 18-year research path to be able to more accurately determine how each of the curcuminoids in a curcumin extract, there are 3 principal curcuminoids in a curcumin extract, how each of them contribute to this polypharmacology, and then why, when combined in a way where the BDMC is dominant, why it's more effective in the resolution of the symptoms, more effective than regular curcumin that comes off the line after extraction with a very low concentration of BDMC in it. A regular curcumin has a BDMC concentration of about 1 to 2% concentration. BDMC stands for bis-dimethoxycurcumin. And in regular curcumin, you have diferrylmethane, you have dimethoxycurcumin, and then you have bis-dimethoxycurcumin, which again only exists at approximately 1 and maybe as high as 3% concentration. What we've endeavored to do then is to isolate the curcuminoids and study them individually, as I mentioned earlier, and determine which drug targets in our cells which proteins in our cells they neutralize in the context of their anti-inflammatory activity. It has been assumed for the longest time, for decades and decades, that all three curcuminoids have analogous activity. They have very similar activity, but it's not the case. What we showed and what has been publicly and published in peer-reviewed journals now from our facility, what we showed was that the BDMC has a specific target that it can neutralize that the other curcuminoids cannot neutralize. And that's called a kinase. It's an MSK1 is the specific kinase. And it is what lends the overall composition of the curcumin BioBDMC, the unique activity that serves the patient with a more thorough anti-inflammatory effect for chronic conditions. It works more expeditiously within 2 hours rather than 2 days. The reason is that this MSK is an activation kinase. It turns on, like a key, turns on the transcription of cytokines from or by NF-kappa B. And so most people aren't really aware of the mechanism of how curcumin works. If you're involved in research and clinical applications like you are, Lauren, you're probably well aware that curcumin in general can inhibit NF-kappa B, which is a transcription factor that makes or transcribes from our genes all the cytokines that we know elicit an inflammatory response, like interleukin-1, interleukin-2, interleukin-3, interleukin-8. I can go on and on. TNF-alpha, people are probably aware of. And those interleukins serve as proteins and beacons and messengers to beacon the immune system to come to the site at which these interleukins are being transcribed. And that site is the site of injury, infection, things like that. And the immune system comes in and itself starts to secrete these compounds to create an effect that's we call immunity and we call inflammation. And so all the curcuminoids can inhibit NF-B, but they inhibit it so that it slows it down. It slows down its ability to make these cytokines, but it takes a few days for that activity to be apparent. But when we inhibit MSK1 with the curcumin 3 element plus the NF-kappa B, the effect is noticeable within an hour or so, because we're no longer waiting to slow down the movement of the NF-kappa B. We're actually turning it off. So it doesn't matter how much of it gets into the nucleus to the genetic material to make our cytokines. We're turning it off so that it's not effective in actually transcribing the cytokines after an inflammatory action. And that's how this compound works in the context of inflammation. But there's so many more levels of activity for BDMC that we've discovered that we've begun to understand that it can play a role in the curcumin complex that's more profound in terms of the polypharmacology when we raise its concentration, more profound than curcumin on its own. The interesting thing is that the BDMC on its own is very limited with how it can work. And here comes into play our holistic approach that is required with the other curcuminoids available in the context of the BDMC existence.
Franco, I know the product curcumin 3 has all 3 curcuminoids in it, and the curcumin 3 has 50% of the capsule is made up of BDMC. Why do we need all the other curcumins in there? Why wouldn't we just use the BDMC directly?
Well, first of all, BDMC on its own is a little bit harder to extract at full concentration because it exists at such a low concentration. It creates a little bit of a financial or economic limitation. Nevertheless, when you look at the science, it's not worth having the BDMC on its own because it's very targeted in its activity. We talked earlier about the allopathic medical objective of targeted medicine. In neutralizing MSK1, for example, like the BDMC can, we do get an effect of an anti-inflammatory effect. But by neutralizing both NF-kappa B and the MSK1, the effect is compounded because what we're doing is we're neutralizing 2 proteins in the same pathway So this compounding the inhibition of that pathway's progression.
So
That pathway is called the NF-kappa B signaling pathway, and it is central to every single inflammatory cascade we experience. And in fact, we know that inflammation is the core chemistry of every disease. The different drugs we might use, the different whether they're allopathic or herbal, they each will target different proteins in that pathway depending on the type of disease and where it needs to be neutralized.
So how would my patients benefit by using curcumin 3 and higher dosages of BDMC? And what would we be expecting in clinical practice?
Well, the one thing that is definitely now apparent, and wasn't apparent up to about 7 years ago, is that the speed of activity for the curcumin 3 combined curcuminoids with a higher level of BDMC, the speed of activity occurs within a couple of hours, as opposed to a couple of days for a lot of these conditions. The other thing that happens is that for chronic conditions that are more systemic, like an autoimmune condition, the curcumin 3 complex works much better because we're neutralizing that MSK again. And MSK has been recognized to be dysregulated or more active in conditions like autoimmune conditions and cancers, in drug-resistant cancers in particular, where MSK is dysregulated. And so we will see a more reliable therapeutic outcome using the curcumin 3, which has the higher BDMC level in it.
Okay, great. And I've noticed that clinically as well. I've used it on a variety of conditions, and I find that it seems to activate much quicker. And in the more complex cases, I find that even in those autoimmune conditions and stuff where you don't expect curcumin to respond until maybe months of therapy, do you see people starting to feel better? This could be weeks. And there is something about the speed of its activity, for sure, that we're witnessing clinically. There's a lot made about curcumin and curcuminoids absorption, whether it's digestive degradation, liver degradation, P450 pathway breakdown. What makes your product, your technology, with the augmentation of curcumin 3, the third curcuminoid, BDMC, not see that? What makes it work so much better without having that degradation effect happening in the gut?
Well, I think, consumers have been well educated by manufacturers of curcumin about the bioavailability limitations. And my opinion, based on our long-standing research, is that this bioavailability limitation is over-exaggerated. And the reason why it's exaggerated is because a lot of these companies try to market their specific proprietary compensatory action to bioavailability, meaning we have phytosome activity that creates a micelle that improves solubility or improves suspensibility. So curcumin is hydrophobic, which means it's not soluble unless it's assisted by something like a phytosome, like phosphatidylcholine. And then we've got a lot of companies adding black pepper extract, piperine, and claiming that improves the bioavailability. However, what we've seen with our compound, where we've escalated the amount of BDMC in the composition, is that two things happen. One, BDMC has an ORAC value that's just astronomical. The antioxidant activity is very, very high. So it protects the complex, the entire complex, from oxidation or auto-oxidation. So there's two levels of breakdown, if you want to call it, or oxidation or catabolism of the curcuminoid that occur upon consumption. One would be the auto degradation. That would occur if you leave it out in a glass, if you leave it as it's transiting through your intestinal tract. And then you have enzymatic activity that's caused by the metabolism. Your liver's role is to try and eliminate things that end up in your bloodstream that they feels is not supposed to be there. And this P450 enzyme, these CYP enzymes that you referred to, are part of the process of degradation to remove the compounds from your blood. This is how drugs are removed. And typically, we see that BDMC actually slows down the enzymes that are responsible for degrading the curcumin in the bloodstream. So what we're doing is we're preserving that curcumin and all of the curcuminoids in the context of this high ORAC and protective feature of BDMC, so that they survive longer in the bloodstream and that they survive the autodegradative activity.
Is BDMC therapy effective in conditions like cancer, autoimmunity, some of these more complex conditions? I know curcumin is widely used in various forms of alternative or supportive cancer therapy, breast cancer. What do you know about that, and what have you seen in the research or researched in supporting using this product in cancer therapy and complex conditions, autoimmune conditions?
Well, you know, we have not done clinical work in cancer, but we've done a lot of in vitro work in cancer, and we've shown that, you know, the BDMC50 and BDMC30 that we manufacture definitely has it has a significant effect on glioblastoma, as what's the cancer that we study. We've studied other cancers, and it's similar in its effectiveness. But again, we haven't done this clinically. So there is research in the literature showing that BDMC can be used to treat various cancers clinically. And, you know, we've been fortunate enough to be engaged in this research and have been able to discover the mechanism. The mechanism being the inhibition of MSK. And in fact, the pharmaceutical industry is in pursuit of MSK inhibitors because they know that in some drugs that recur after they've been treated with a chemotherapeutic substance successfully, they usually recur with an elevated activity of MSK1, and neutralizing MSK1 resuscitates the chemotherapeutic drug. So what we've been able to show is the mechanism by which it works. We've been able to show in some cancers in vitro how it works profoundly to induce apoptosis, destroying the cancer cells. So there's a place in cancer, there's a place in inflammation, autoimmunity. But what's very important is that the other curcuminoids need to be also in the composition in order for it to have optimal effects. Because remember, one of the strategies even for cancer inhibition is the inhibition of NF-kappa B. So stopping NF-kappa B is important in the context of these cancers, but stopping both MSK1 and NF-kappa B delivers the biggest impact. So
what is the BDMC technology? What is the BDMC50 technology? What does it do? How is it done? What creates the therapeutic effect when we take it? Like, how does it compare to standard curcumin?
Well, standard curcumin is not able to inhibit the MSK1. We've shown with multiple brands that are out in the marketplace, multiple sources from different suppliers, it cannot inhibit MSK1. Our curcumin BioBDMC, either the BioBDMC30, but especially the BioBDMC50, which is the clinical application, will inhibit MSK1. And that's the differential, because once you inhibit MSK1, you have a more expeditious activity. You have an ability then for the drug to work in conditions that are resistant to drug use application. You have now a curcumin that can work in autoimmune conditions where regular curcumin, I know I don't even have to ask, but I know in your clinical practice, regular curcumin is not very effective for autoimmune conditions. People may take the edge off it, but it's just not really giving the impact that changes people's lives the way the BioBDMC version will.
Great. And I know there's a lot of research also in the neurological application of BioBDMC50. What's your experience in that area?
It's amazing, you know. So again, you know, we're just on the cusp now of entering the clinical trials that will show a lot of new ways the drug can be used. And I call it a drug, it's a natural drug. But from a neurological standpoint, it's amazing. You know, we know the curcuminoids will cross the blood-brain barrier. And the BDMC component is the smallest curcuminoid, which can cross even more easily. But the interesting thing is in the scope of Alzheimer's and the amyloid plaque development in Alzheimer's. Now, there's a lot of controversy over whether or not the amyloid plaque is a factor that contributes to the end pathology of Alzheimer's, or the plaque is a consequence of the pathology. And it's interesting because we've shown in our research, and I have 2 papers published in Elsevier Medical Journal, that that plaque is loaded usually with heavy metals, and we wonder whether or not in the research paper I discuss whether or not that plaque is a deposit of metals, whether or not the amyloid protein itself is a chelating agent that chelates our metals and then brings them into play into the plaque. The endpoint that causes cell death or apoptosis in Alzheimer's is the delineation of what's called the tau protein, which comes as a secondary stage. So, but having said that, what's really interesting is that the BioBDMC component will inhibit what's called BASE1. BASE1 is an enzyme designed to actually make, it's one of the rate-limiting enzymes for the generation of the amyloid protein. So you can inhibit that enzyme with curcumin. The other thing that's been shown in research is that curcumin will also get in the way of the amyloid proteins aggregating so that they don't generate or make the plaque. The other thing that's really cool about this curcumin is now it's shown that it's also a chelating agent for heavy metals. So when it comes to preserving neurological health, there's so many elements to the curcumin that we're talking about. Curcumin BioBDMC will inhibit BASE1 activity so that you have the amyloid, you have a lower rate of amyloid plaque accumulation. You have an antioxidant activity that preserves that brain. You have an anti-inflammatory activity that slows down the inflammation that's associated with Alzheimer's, and a potential to reduce the rate in which the pathology advances.
So how safe is it? I know if you take 1 capsule of CURCUMIN III, that's 25 times more BDMC than you would find, say, in a standard curcumin capsule, standard 95% standard curcumin. How safe is taking that quantity of BDMC?
Well, in terms of the research and the mechanism of activity, we have no reason to believe that there are any significant adverse events that would be any different than regular curcumin. You know, BioBDMC itself, you know, has been up to about 3% concentration in curcumin for 2,000 years. Is it toxic? You know, we'd say not. We've had research submitted to Health Canada that proves safety based on the safety research that we supply. And we've been in the marketplace for 4 years with the technology with no adverse events in the general population. So I'd be comfortable saying that, you know, we're looking at a natural drug that's as safe as the safest.
Great. Okay, Franco, did we cover everything? Is there anything else you want to cover?
I think you covered everything there.
We can edit this part out. We can edit out some stuff.
Yeah.
Any last things we should get you on tape with?
Well, you know, I think it's really important for people to understand that, you know, inflammation is required for recovery, yet prolonged inflammation can be inhibitive of recovery and cause general damage. And I think what has to be also understood is that as we age, our ability to regulate that inflammatory activity declines. It's one of the reasons why we can't engage in the same level of physical intensity and then the next day feel as good as we would have when we were younger. We have that stiffness. And so I think, you know, as much as these products, the curcumin BioBDMC is a therapeutic product, I think as we get older, it's also something we should consider as a prophylactic to kind of offset that general escalation of subclinical inflammation. You know, 1 capsule a day. I mean, because I have ulcerative colitis, I take 5 to 6 capsules a day. You know, 3 to 4, which I consider the therapeutic, and a couple of extra just for that, you know, aging factor. And it's kept me at age 70. No, I'm joking. Age 54, feeling young. You know, and if I don't take it.
Yeah. So Franco, I know inflammation is a double-edged sword because we need it to heal. And if you look at a lot of the research, many of the chronic degenerative conditions have an out of control or an imbalanced inflammatory response occurring, which is
Yes.
part of the degeneration that we see in those conditions. Is curcumin 3 a product that one might want to consider taking a capsule or 2 a day on a regular basis?
Absolutely. Yeah, for sure. Especially after age 30, 35. You know, it's kind of that countermeasure that kind of, it allows you an opportunity to function from an inflammatory standpoint like you were younger. I mean, there's so many diseases and disorders that including aging. Aging is a function of disarray in our cells, and it's a function of oxidation and inflammation.
Yes, Franco, do you find that BDMC, is it a drug? Is it a natural substance? Is it both? How would you define it in the way that you've presented it?
That's a great question. So I mean, it's obviously a natural compound. So, you know, I call it a natural medicine or a natural drug. And the reason is that its activity is as potent as powerful drugs, yet, you know, it's a natural constituent. And the nice thing is that unlike a lot of these potent drugs, you don't see the side effects and the adverse events that might be associated with them. So I think it's a great first round treatment for most inflammatory diseases that poses lowest risk right now in terms of even in terms of non-steroidal anti-inflammatories, which we seem to think are benign in terms of their adverse and secondary effects. But, you know, it's clearly recognizable today that there are a lot of mucosal membrane or gastrointestinal ailments that rise out of chronic use of those NSAIDs. And so this is a great alternative that doesn't have those adverse events, those adverse risks.
I'm surprised that Big Pharma hasn't caught on to this.
Well, there's various. Big Pharma hasn't caught on to the BDMC activity. Yeah, you know, the thing is that Big Pharma is more apt to looking for unnatural analogs, and that's what their model is. And I think that's where the adverse events come when you have unnatural analogs of these compounds.
Franco, fantastic. We really appreciate you joining us today here and going through curcumin 3 therapy. Really appreciate your time and effort and everything that you've done in pushing forward new technology. And I know this isn't the last time we're going to be speaking with you. Great job. Thanks for joining us. Have a great day.
Lauren, thanks for everything you do. And it's been great talking to you and working with you.